Hello student, I am Dr. Manjusha Vijayengole, Head Department of Botany, Kisanveer Mahavidyalaya, Vai. In this presentation, we have to study mitochondria, which belongs to semester first, paper second of B.Sc part first. And the title of paper is Biomolecules and Cell Biology. Popularly, mitochondria is known as a powerhouse of a cell. All of you know that powerhouse means at that site where energy is stored. In these cell organelle, energy is stored in the form of ATP molecules, and therefore these mitochondria in a singular form it is known as a mitochondrion, which is a double membrane bounded organelle and which is found in eukaryotic cells. These are found inside the cytoplasm and these are essential in the cell digestive system. They play major role in breaking of the nutrients and generating the energy rich molecules in the form of ATP for the cell. Many of the biochemical reaction which is involved in the cellular respirations, these all reactions, these are takes place within the mitochondria. The term mitochondria is derived from the Greek words mitos and chondrion. These are the two different splitting of that, and which means thread and granules like. Means meaning of that mitochondria is thread and granules like. It was first described by German pathologist, and name of the German pathologist is Richard Altman in the year 1890. The definition of mitochondria, this definition of mitochondria is it is a double membrane system, double membrane organelles which is present in the cytoplasm of all eukaryotic cells, and these are responsible for the production of energy rich molecules in the form of ATP, that is nothing but adenosine triphosphate. These ATP molecules, these are the main energy molecules which is used by that cell. When we observe the structure of that mitochondria, these are nothing but a double membrane system, outer membrane, inner membrane. In between these two membrane space is present. Then inside that we observe foldings, and in that the matrix region is present. The detailed ultrastructure we have to see in the next plate. But the length of this mitochondria is about 1 to 2 micron and diameter is about 0.1 to 0.5 micron. In this ultrastructure, we found that these mitochondria, it is a double membrane system, outer membrane and inner membrane. The chemical composition of that two membrane, these are different one, but this outer membrane is permeable, and in between these two membrane, we observe the space is present, and that space is termed as intermembrane space. Then from that inner membrane, we observe certain expansion in the form of foldings, and that folding is responsible for giving more and more space for that cellular respiration, and that foldings, these are termed as a cristae. And in the inside that mitochondrial inner membrane, the granular structure is present, that is termed as a matrix. In that matrix, the mitochondrial ribosomes, these are present. This is nothing but ribosomes which are joined with each other, is mainly responsible for protein synthesis. It is joined on the mRNA molecules. Then in the mitochondria, their own genetic material is present in the form of a DNA, and that is termed as a mitochondrial DNA. On these foldings, that folding is termed as a cristae, and that cristae, these are mainly playing important role in electron transfer system as well as oxidative phosphorylation as well as the steroid metabolism. Then when we observe this single outgrowth, that is a cristae present on that foldings of that inner membrane, these cristae, these are having the inner membrane, outer membrane, in between them the peripheral space is present, and in a central head region we found that there is intracrystal space is present. Then on that we observe certain outgrowths, these are present. These outgrowth having the stalk, that stalk is termed as F0, and the head region which is present, which is a spherical shaped, spear shaped, it is termed as F1. And these are mainly attached to that crystal membrane. This F0 stalk and F1, that spear-like body, these are mainly responsible for, or on that, the enzyme which is present, these are ATP synthetase, means it is playing important role in actual ATP synthesis. The mitochondria is a double membrane, rod-shaped structure found in both plant as well as animal cells. The size ranges from 0.1 to 1 micrometer in a diameter. The structure, these are having the outer membrane and inner membrane, and a gel-like the protoplast material is present, which is termed as a matrix. The outer membrane and the inner membrane are made up of proteins and phospholipids layer. These are separated from each other by the space which is termed as the intermembrane space. The outer membrane covers the surface of mitochondria, and these are having the larger number of special protein which is termed as a porins. It is a freely permeable to ions, nutrients, nutrient molecules, energy molecules in the form of ADP molecules as well as ATP molecules. When we observe these different layers of that mitochondria, these different membrane of a mitochondria, these are having a different type of enzymes. I mean, for example, outer membrane having the NADH cytochrome, B3 reductase, as well as inner membrane having succinate dehydrogenase. In between the space present in between these two membrane, that is having nucleoside monophosphokinase. In a matrix region, the number of enzyme present which is responsible for Krebs cycle, means such a type of a variety of enzyme present on the outer membrane, inner membrane, the space which is present in between two membrane, the matrix region, etc. of these mitochondria. Then function of a mitochondria. The students, you remember that mitochondria is termed as a powerhouse of cell, means in that from that food molecules, their cellular respiration is that takes place, and it results into formation of energy rich molecules in the form of ATP, that is nothing but adenosine triphosphate. It is the main function of a mitochondria, but how it is that takes place inside that mitochondria in this diagrammatic representation, I show you that this mitochondria is regarded as the powerhouse of the cell, and it is the site of a respiration. The general formula for the glucose oxidation is in this manner. Glucose means C6H12O6. Their oxidation is that takes place by 6 molecule of oxygen, and it results into formation of a 6 molecule of a carbon dioxide, 6 molecules of water, and it results into production of energy in the form of 686 kilocalories. Glucose is degraded into 2 pyruvate molecules. All of you know that these 2 pyruvate molecules, it is produced in the process which is known as a glycolysis. Means in a glycolysis, from single glucose molecule, 2 pyruvate molecules these are produced. Then the oxidation of a pyruvate is takes place in the mitochondria, and it is converted into formation of acetyl-CoA, which is termed as acetyl coenzyme A. Then that acetyl coenzyme A is again metabolized through one cycle, and it is known as a Krebs cycle. It is also Krebs cycle. It is also known as a citric acid cycle or tricarboxylic acid cycle. Dear students, all of you know this glycolysis, Krebs cycle, TCA cycle in 12th standard. In this cycle, energy is liberated and carbon dioxide is produced, and there in this process, the major form which is seen, that is reduction of coenzyme NADH and FADH2, and it results into formation of energy rich product in the form of ATP molecule, and the formation of ATP molecule is takes place. That process is termed as a oxidative phosphorylation. Then here there are 3 complexes which is involved in that process of a formation of a ATP molecule: complex 1, complex 2, complex 3, complex 4. These are all complexes. These are responsible for the complete oxidation of one glucose molecule, and it results into formation of 6 carbon dioxide molecules, 6 water molecules, and in addition to them, the energy is maximum amount of energy is produced, and it results into formation of 36 molecules of ATP. These are produced from that single glucose molecule in these mitochondria. Thank you very much.